Learning outcomes. After studying this module, you shall be able to conceptualize personality as in early physiological approaches, learn about the physiological measures of personality, evaluate the physiological based personality theories. Introduction. The philosophical traditions since the time immemorial are intrigued by the mind-body problem. This debate that has been passed down the centuries tries to narrow down the physical and psychological experiences of humans. While a league of people believe in the independent existence of the two, another large group of people research the codependence and coexistence of the mind and the body. In wake of modern technologies, a tremendous shift of focus has been observed while addressing the biological basis of psychological constructs. Many modern psychologists believe that differences in bodily processes result in differences in personality. The roots of this tradition of highlighting physiology in personality can be traced back to Hippocrates in the four temperaments. According to the Hippocrates, personality is formulated through combination of the four body humours, namely blood, yellow bile, black bile and phlegm. His proto-psychological theory suggests that there are fundamentally four types of personality types, namely sanguine, phlegmatic, choleric and melancholic. William Herbert Sheldon pioneered in establishing the biological basis of personality. He developed somatypes to categorize individuals based on their physiques. This approach to personality comes under the type theories. Having studied a large number of people, Sheldon narrowed down to three broad types of personalities. Ectomorphic, characterized as linear, thin, usually tall, fragile, lightly muscled, flat-chested and delicate. Described as cerebrotonic, inclined to desire isolation, solitude and concealment and being tense, anxious, restrained in posture and movement, introverted and secretive. Mesomorphic, characterized as hard, rugged, triangular, athletically built with well developed muscles, thick skin and good posture, described as somatonic, inclined towards physical adventure and risk taking and being vigorous, courageous, assertive, direct and dominant. Endomorphic, characterized as round, usually short and soft with underdeveloped muscles and having difficulty losing weight, described as visitonic, enjoying food, people and affection, having slow reactions and being disposed to complacency. Typology of personalities can also be found in ancient Indian philosophies. The Sankhya school of Hindu philosophy defines three gunas or virtues. Dominance and balance of these gunas are manifested as what we call as personality in modern times. However, according to this tradition, nothing is cent% defines by only one type of virtue. Human behavior characterization according to this is as follows. Sattva is the quality of balance, harmony, goodness, purity, universalizing, holistic, constructive, creative, building, positive, peaceful and virtuous. Rajas is the quality of passion, activity, neither good nor bad and sometimes either, self-centeredness, egoistic, individualizing, driven moving, dynamic. Tamas is the quality of imbalance, disorder, chaos, anxiety, impure, destructive, delusion, negative, dull or inactive, apathy, inertia or lethargy, violent, vicious, ignorant. Physiological measures in personality research. Drawing on the above, many personality researchers employ scientific and empirical physiological means to gather insights deep onto the construct of personality. Most commonly used among the methods is the use of electrodes or sensors placed on the participant's skin. Other and more complex methods include measurement of cardiovascular and brain activities. The rationale behind exercising such methods is again attributable to the fact that personality is a sum of physiological patterns and activates through the body. However, in light of various researches, it is difficult to conclusively comment whether physiology causes psychology. Nonetheless, there is definitely a strong correlation between the two. The following section describes the above mentioned methods in detail. Galvanic skin response. Electrodermal activity of measurement of skin. Conductance enables researchers to directly measure sympathetic nervous system activity. Our skin contains numerous sweat glands, especially in hands and paws. These sweat glands are directly influenced by sympathetic nervous system operating under the autonomic nervous system, which prepares an individual for flight or fight response. In presence of a stressor, the sympathetic nervous system directs sweat glands to produce sweat. If the stimulation is prolonged, sweat pours out from our palms. That is why we have the proverb sweaty palms. Even before the sweat appears, it is possible to measure sympathetic response by running a minute amount of current through the skin. Since water conducts electricity, in this case sweat, higher the concentration of sweat, easier is the conductance, measured through a galvanometer. Thus, applying this logic, scientists measure electrodermal activity of research participants. Technically, in this procedure, two electrodes are attached to the hand. A very low voltage current is put through one electrode and it is observed that how much of the electricity reaches the other. The difference in the amount of electricity in both the electrodes indicates how well the skin conducted. The more sympathetic activity, greater is the skin response. Interestingly, this measure is so precise that the participant does not even feel the passing of electricity through the skin. Almost all kinds of stimuli like sudden noise, pinprick, emotionally charged or conditioned stimulus can elicit a skin response. What interests psychologists is observation of skin conductance in absence of external stimuli. Let us discuss an example in this context. Imagine a person who is placed in a relaxed and calm setting, sans any threat or explicit external stimuli to cause disturbance. Galvanic skin response under this setting should ideally be very less, as the sympathetic activity is expected to be low. However, if the person exhibits spontaneous electrodermal response, even though there is nothing that objectively stimulating, it can be thought of as reflection of personality traits. Neuroticism or anxieties for the matter are traits that are associated with non-specific skin conductance. By Kruse and Larson in 1994, a person who is rated high on anxiety has chronic sympathetic nervous system activation. Similarly, other traits and differences among people can be measured through galvanic skin response. Cardiovascular activity. The cardiovascular system contains of the heart and blood vessels. A complex system that it is, its measurement is multifaceted task as well. One can look at the pressure exerted by blood on the walls of the vessels or measure the electrical conductance of the heart. There are many methods through which one can monitor the cardiovascular functioning. Some major ones are discussed below. Blood reports. Blood tests check the level of certain fats, cholesterol, sugar and proteins in your blood. Abnormal levels may be a sign that you are at risk of CHD. A cholesterol test, also called a lipid panel or lipid profile, measures the fats, lipids in your blood. The measurement can indicate the risk of having a heart attack or other heart disease. The test typically includes measurement of total cholesterol. This is the sum of blood's cholesterol content. A high level can lead to an increased risk of heart disease. Ideally, total cholesterol should be below 200 milligrams per deciliter or 5.2 millimoles per liter. Low density lipoprotein LDL cholesterol. This is sometimes called the bad cholesterol. Too much of it in the blood causes the accumulation of fatty deposits, plaques, in your arteries, which reduces blood flow. These plaques sometimes rupture and lead to major heart and vascular problems. Ideally, LDL cholesterol levels should be less than 130 mg per dl, and under 100 mg per dl is even better. High density lipoprotein HDL cholesterol. This is sometimes called the good cholesterol because it helps carry away the LDL cholesterol, keeping arteries open and the blood flowing more freely. Ideally, HDL cholesterol level should be 60 mg per deciliter or higher, though it is common that HDL cholesterol is higher in women than men. Triglycerides. Triglycerides are another type of fat in the blood. High triglyceride level indicate that the amounts of calories regularly consumed are more than what is burnt. High levels increase the heart risk. Ideally, triglyceride level should be less than 150 milligram per deciliter. The American Heart Association AHA recommends that a high triglyceride level of 100 milligram per deciliter or lower is considered optimal. The blood can also be tested for proteins like fibrinogen, which encourages blood clotting, but high levels of which are linked to an increased risk of heart attack and vascular disease. Another protein is N-terminal pro-B type natriuretic peptide NT-proBNP, or the non-traditional blood protein made in the heart and found in the blood. High levels are associated with increased risk of cardiovascular disease, heart attack and heart failure development. Elevated levels are associated with development of heart failure and worse prognosis. Sphygmomanometer. It is an instrument for measuring blood pressure, typically consisting of an inflatable rubber cuff which is applied to the arm and connected to the column of mercury next to a graduated scale, enabling the determination of systolic and diastolic blood pressure by increasing and gradually releasing the pressure in the cuff. Even quite small increases in the blood pressure, if they persist, can have significant effects. For example, an average increase in the population of 2 mmHg is systolic blood pressure increases the death rate from stroke by 10% and from coronary heart diseases by 7%. Since hypertension is one of the risk factors of CHD, measuring blood pressure might assist diagnosis of CHD. Brain activity. Brain, the master of all systems of the body, consistently produces and conducts small amounts of electricity. These electrical messages can easily be measured through the electroencephalogram EEG by placing electrodes on the scalp. The EEG can record brain activity for several regions of the brain in various contexts like sleep or hyperactivity. Due to this regional specification of activities that an EEG can provide is very useful for ascertaining parts of the brain with their associated activities. For example, Forebrain region is linked to cognitive activities and the information processing. In context of personality, researchers are interested in measuring brain activities of typical patterns of behaviors such as extroversion or introversion. Another way of utilizing EEG is through evoked potential measurement. That is, recording brain waves in response to a particular stimulus such as sudden flashing of light to the eyes. This indicates the participant's readiness or responsiveness, further adding to our understanding of personality differences. More recent and powerful techniques to record brain activities have been developed. Positron emission tomography (PET) and functional magnetic resonance imaging (fMRI) are non-invasive techniques used for mapping the structure and the function of the brain. fMRI specially has become an eye candy of biopsychologists. Advanced and in-depth information for various psychoclinical conditions such as depression and schizophrenia have been gained through the employment of this technique. An important study was published by Canley et al. In 2001 in which they used fMRI technique to scan the brains of people as they looked at 20 negative pictures, spiders, people crying, etc., and 20 positive pictures like cute puppies, flowers, etc. It was found that different regions responded differently to negative and positive images. Moreover, strong correlation between personality and emotional response was observed. Specifically, neuroticism corresponded to the increased frontal brain activation in response to negative images, while extroverted people showed increased brain activities in the frontal region in response to positive images. Brain imaging techniques have really revolutionized researches in this direction. Physiological theories of personality. The following sections describe some of the leading physiological theories of personality. Physiological approach to the personality, as it might be clear by now, highlight and proposes biological explanation to individual differences and personality characteristics. Extraversion-Introversion. Do you like to tell jokes and funny stories to your friends, or do you prefer to spend a quiet holiday fishing by the lake? If you relate more to the former, you are more likely to be labeled as an extroverted person, whereas people who are reserved and like to keep aloof from others are referred to as introverted. Extraversion-Introversion dimensions of personality has caught attention of many psychologists, including Carl Jung. Among those who have discussed this facet of personality, H.J. Eysenck emerges to be the leader, having provided biological reasons of why extroverted people are different from introverted personalities. Introverted people prefer quiet activities such as reading as compared to extroverts who prefer high stimulating activities seeking other people's company. In his book, The Biological Basis of Personality, Eysenck in 1967 proposes that introverted people are characterized by higher levels of activity in the brain's ascending reticular activating system (ARAS) than extroverts. The ARAS located in the brainstem controls the overall cortical arousal. ARAS in the 1960s was thought to be as a gate through which messages entered the cortex. According to Eysenck, higher activity in introverts indicates that they have higher resting levels of cortical arousal. That is, ARAS lets in too much stimulation. Therefore, such people prefer activities that are quiet and seek less stimulation. On the contrary, extroverts have little resting levels and stimulation, which they compensate by engaging in behaviors that increase their level of arousal. Sensation seeking. Remember the last time you went down a roller coaster? How was the experience? Did you feel a rush? Probably you noticed your pounding heart. The thrill and sensation evoked by these activities is massive. What might have been a one-time experience for you is a regular for some. Sensation seeking is another dimension of personality that has strong physiological basis. It is the tendency to seek thrilling and exciting activities, to take risks and to avoid boredom. Sensation seeking personalities seek super thrilling activities such as the parkour or taking risky selfies. Research around sensation seeking developed out of the researches for sensation deprivation by Hebb in 1955. Hebb developed theory of optimal arousal, which was later incorporated by Eysenck in his theory of extraversion. According to this theory, people are motivated to reach an optimal level of arousal, a level that is just ideal. If people are aroused less than this level, they tend to increase their arousal, that is rewarding. On the other hand, if they are aroused over the optimal level, then decrease in arousal is rewarding. However, in light to various theories of motivation, example drive theory, motivation involves reduction and not elevation of tension. Reducing the tension is the goal, which is in contradiction to Hebb's proposal of sensation seeking. However, his behavior is consistent with the several sensory deprivation researches by Zuckerman in 1978. The nervous system seems to require some amount of sensory input. Zuckerman and Haber in 1965 in their early experiments on sensory deprivation found that some people more than others are easily distressed in absence of sensation. These were the ones who quit the study early or requested lots of sensory materials, reading materials, tapes. During this experiment, they were identified as persons who had high need for sensations, as they were least tolerant of sensory deprivation. They reported feelings of unpleasantness under deprived circumstances. Based on his observations, Zuckerman in 1978 developed a questionnaire to assess people's need for sensation seeking. He defined the following facets characterizing people with high sensation seeking tendencies: thrill and adventure seeking, experience seeking, boredom susceptibility, and disinhibition. According to him, preference of people determined on the basis of test items on sensation seeking scale predicted how well people tolerated sensory deprivation sessions. Skydivers, bungee jumpers, formula car racers are likely to score high on this scale. According to Zuckerman, there is a physiological basis for sensation seeking tendencies. More recently, his researches have emphasized the role of neurotransmitters as an explanation for sensation seeking personality. As one may recall, neurotransmitters are chemicals stored in axon terminals of neurons that carry forward the neural messages across synapse, the cleft between neuron and its follower. Though present in minuscule quantities, levels of neurotransmitter can have major behavioral and psychological effects. Thus, the levels of neurotransmitters strictly need to be monitored. Certain enzymes such as the monoamine oxidase MAO help maintaining this balance. Excessive amount of MAO would break down too much of neurotransmitters, while too less of it would allow excessive neurotransmission. Especially if the quantity of MAO is very low, it may cause jittery movements. On the other hand, overactivity of the enzyme would result in clumsy, dull sensation. High sensation seekers tend to have low levels of MAO enzymes by Zuckerman in 1991. Low levels indicate that there is less nervous inhibition and availability of more neurotransmitters may cause sensation-seeking behavior. Morningness-Eveningness. People often characterize themselves as larks and owls. Difference in the morning and evening type of people can be perhaps attributed to the underlying biological circadian rhythm. Neurotransmitters and personality. Neurotransmitters are chemicals in the nerve cells that are responsible for the transmission of nerve impulses across synaptic cleft. In recent times, the role of neurotransmitters is gaining importance so as to explain the individual differences among people. Dopamine appears to be associated with pleasure. While traces of dopamine can be found all over the brain, its transmission occurs majorly in the mesocorticolimbic pathway. This pathway originates from the ventral tegmental area in the midbrain and extends to the forebrain. Drug abuse, such as consumption of cocaine, mimic dopamine, depletes body of its natural synthesis of the chemical, hence leading feelings of unpleasantness, therefore creating a drive or urge to obtain more of the drug. Serotonin is another neurotransmitter that has received much attention in the light of its relevance in clinical depression. Another neurotransmitter, norepinephrine, is involved in activating the sympathetic nervous system. Tri-dimensional personality theory given by Cloninger in 1986 suggests that these three neurotransmitters play a vital role in personality traits. For example, novelty seeking was found to be associated with low levels of dopamine, while harm avoidance is one behavior that was observed to be released to abnormalities in serotonin level. Norepinephrine, on the other hand, is associated with pleasure dependence. Low levels of this chemical can be seen in people who are highly motivated to work for long and with persistence. Limitations. Theories within the physiological approach support nature over nurture. Be that as it may, it is restricting to depict behavior exclusively as a product of either nature or nurture and endeavors to do this. Think little of the unpredictability of human behavior. It is more probable that behavior is because of an association between nature, science and support environment. A constraint that is most physiological approaches are reductionist and do not give enough data to completely clarify human behavior. People might be inclined to specific practices, yet these practices may not be shown unless they are activated by variables in the environment. This is known as diathesis stress model of human behavior. Cultural and social factors too seem to play a vital role in shaping personalities and creating a sense of self-identity. Those nuances where the culture underline the character of human beings are not addressed in this approach, which is fairly universal. Summary. Physiological approach to personality began on the lines of Hippocrates' work, where he suggests that personality is formulated through combination of four body humours, namely blood, yellow bile, black bile and phlegm. His proto-psychological theory suggests that there are fundamentally four types of personality, namely sanguine, phlegmatic, choleric and melancholic. Sheldon in modern times suggested three typologies. He developed soma types to categorize individuals based on their physiques. This approach to personality comes under the type theories. Technological advancement has enabled modern man to directly measure physiological correlates of personality. Electrodermal activity of measurement of skin conductance enables researchers to directly measure sympathetic nervous system activity. There are many methods through which one can monitor the cardiovascular functioning. For example, blood cholesterol measure or through Sphygmomanometer measure of brain activity include EEG, PET scan, MRI, fMRI, etc. The EEG can record brain activity for several regions of the brain in various contexts like sleep and hyperactivity. Positron emission tomography (PET) and functional magnetic resonance imaging (fMRI) are non-invasive techniques used for mapping the structure and function of the brain. fMRI especially has become an eye candy for biopsychologists. Theoretical frameworks of physiological personality provide a wide spectrum and multiple viewpoints. Eysenck, for example, talked about the biological basis of the two essential nature of introversion and extraversion. He proposes that introverted people are characterized by higher level of activity in the brain, ascending reticular activating system, than extroverts. Zuckerman, on the other hand, emphasized the role of neurotransmitters in sensation seeking. Biological rhythms controlled by the pineal glands of the brain result into various patterns of personality. A strength of the biological approach is that it provides clear predictions for behavior, for example, about the effects of neurotransmitter or the behaviors of people who are genetically related. However, it is limiting to describe behavior solely in terms of either nature or nurture, and attempts to do this underestimate the complexity of human behavior. It is more likely that behavior is due to an interaction between nature, biology and nurture environment.